Okay, up to this point in our course, our biochemistry course, we have studied making proteins from amino acids. We've studied the amino acid sequence, being the primary sequence, folding to secondary and tertiary structure, so that we have the final structure of an enzyme. We examined the activity of an enzyme, the catalytic activity of converting substrate to product. We talked a little bit about some of the experiments that are used to determine the velocity of enzymes. And now, for the next couple sessions, we need to discuss protein methods and how we actually isolate these particular enzymes for study. The enzymes need to be isolated in as pure a form as they can be in order to make sure that we are studying exactly the properties that we intend. So the first step of isolating a protein is that we have to identify where the protein is actually expressed. We can either use individual cells that we culture in a laboratory, or we can use tissues that we can harvest from an animal. Now, the first thing we have to recognize is that the enzyme is typically located inside the cells, which is inside the tissue. So we need to be able to break open the cells in tissues. And this is going to occur by a process called homogenization. And lysis, also called solubilization. So there's several ways to do this. One way is to use sonic disruption. And this is simply using sound waves to break up cells and tissues. I think many of you have seen this type of instrument. These are also used to clean jewelry using sonic measures. A second way is a mild detergent. Now, mild detergents have a property of being able to solubilize cells and tissues by disrupting cell membranes. These disrupt cell membranes by solubilizing the lipid component of the membrane. We'll discuss this a bit later in a later video. Now, one property when we talk about mild detergent is that detergents have a property of being able to disrupt hydrophobic interactions. The cell membranes are typically made up of lipids, which are hydrophobic, and so is the interior of proteins. So if we have a harsh or strong detergent, we could disrupt our enzyme's tertiary structure and eliminate the activity of the enzyme. This is why we want a mild detergent. The detergent needs to be designed so that it can disrupt cell membranes but leave protein structure intact. Another method is called shear force. And this is typically having cells in tissue in a volume of liquid and pushing it rapidly through a small opening with high pressure. The best example of this is a syringe and needle. We would have our cell or tissue material inside the syringe. We have a plunger, and we push down on this plunger and rapidly force the cells through a small gauge needle. This is shear force going through the needle, and this will disrupt the cells and tissue. And finally, the fourth way to disrupt cells is also another type of shear force. But this uses what's called a mortar and pestle. Same type of mortar and pestle that you've seen before. If we have a test tube, this becomes our mortar, and then we have a pestle that fits into the test tube with very high precision on either side of the pestle. As the pestle is plunged into the test tube with our cell material, this material will force itself up through the sides of the pestle, creating a very high shear force. And this will also disrupt cells and tissues. Now, once we do this, what we typically have here is a homogenate. And what is a homogenate? A homogenate is typically cell debris, because the cells and tissues are now lysed open. They're broken open. And so the cell debris is going to contain parts of cells as well as the intracellular organelles, nucleus, mitochondria, lysosomes, endoplasmic reticulum and Golgi, other microsomes, as well as the cytosolic components, the soluble material. So we need to know exactly where our enzyme is located in these different organelles in order to take the next step of protein isolation and purification. Let's say, for example, that our protein is located in the cytosol. The next step we can perform is what's called differential centrifugation. Now, centrifugation is a very simple principle, although the equipment is very sophisticated. We will typically have, for example, a rotor that fits into a centrifuge, spins around a central axis, and we have what are called swinging buckets that are attached to the ends of the rotor. And so what these do is support test tubes. So as the rotor begins to spin, the test tubes move to the outside because of centrifugal force. And this begins to pellet the homogeneous material from the cells and tissue. Now, if we centrifuge at a low RPM, low revolutions per minute, we will pellet large, dense organelles. Because the large, dense organelles have a density that is much greater than the aqueous solution it's in, these will pellet down to the bottom at low speed centrifugation. Which one of the organelles can be considered to be large and dense? Well, this is primarily whole cells that have not been disrupted by our procedures here, and nuclei. Nuclei are very, very large, so that they are very dense. So what we will have is a pellet containing whole cells and nuclei, and we will have a supernatant with everything else. What we can then do is take the supernatant which is the soluble portion, and we can subject that to medium RPMs. So essentially what we're going to do is we're going to spin this rotor faster. At this point, what is going to pellet are smaller, less dense material. And an example of these are mitochondria, lysosomes, endoplasmic reticulum. And of course, we will also have supernatant that is above the pellet. The next thing we can do is take this supernatant and subject this to high RPMs. This will pellet very small, less dense particulates and what are examples of these? These are microsomes. And small membrane pieces that resulted from extensive shear force activity or sonic disruption. And of course, above the pellet, we have supernatant. And finally, we can take this one step further with very high RPMs. At this point, we will be pelleting viruses and large macromolecules. We can even precipitate or pellet large protein complexes with very high RPMs. So this is the principle of differential centrifugation, because simply we are centrifuging at differential speeds. So at low speeds, we are able to pellet large, dense organelles. We can remove these and work with those individually, or take the supernatant, subject that to higher RPMs, and pellet smaller particles. We can isolate these smaller particles and work with them, or take the supernatant and continue spinning these higher and higher centrifugal force. At each successive increase in RPMs or centrifugal force, we will pellet smaller and smaller particles until we are able to actually pellet viruses and large macromolecular complexes. Now at this point, the supernatant is going to contain the cytosol. And we said originally this is where our protein is located. So after doing these successive steps of centrifugation, we can take the supernatant from the very high RPM spin, and this is where our enzyme is. So we've eliminated a lot of the cellular and tissue debris. Now the next step in protein purification is to take the cytosolic fraction, which of course contains a very large number of proteins that are located in the cytosol, and we need to begin fractionating these proteins in here in order to remove the protein of interest and separate it from the other proteins that are not of interest. So the next step we typically do in protein purification is using the salt ammonium sulfate. Ammonium sulfate is able to alter protein solubility. So if we measure this on a graph, protein solubility versus concentration of ammonium sulfate, we will see that a little bit of salt dramatically increases protein solubility. And this is because the salts, dissociated ammonium and sulfate ions, are able to interact with R groups on the amino acids on the surface of the protein. These are forming ionic interactions and adding to protein solubility. So proteins are typically less soluble in pure water. They like a little salt to maintain their solubility. Now eventually what will happen is at certain concentrations of salt, certain proteins will decrease their solubility with increasing salt. What is happening exactly here is that the ammonium sulfate is now interacting with water. Recall that water is surrounding proteins by hydrogen bonding. And when we have excess salt, we are now forming ionic interactions between salt and the water, which removes the water from the protein. If we begin to remove water from protein, the protein begins to aggregate. As it aggregates, the protein precipitates. Now proteins have characteristic solubilities in ammonium sulfate solutions. So as we achieve this concentration of protein, for example, of ammonium sulfate, we will have this protein precipitate out. This may be protein A. Protein A is insoluble at this concentration of ammonium sulfate. As we increase our concentration of ammonium sulfate, we see that there's still proteins that are soluble. However, as we increase the ammonium sulfate, we find another protein group that precipitates at higher concentrations. We can continue this. At even higher concentrations of ammonium sulfate, we will have another group of proteins that will precipitate. So proteins in category A precipitate with low concentrations of ammonium sulfate. Proteins in group C precipitate with high ammonium sulfate. So what we can do is take this precipitated material, remove it from the solution, and then continue. Let's say our protein of interest is in fraction C. As we remove A as a precipitated material, the soluble material, we increase the concentration of ammonium sulfate, and then the proteins in fraction B precipitate. We remove that precipitation, and we take the soluble material and add more sodium sulfate, ammonium sulfate, and then the fractions precipitate in C, which is what we're looking for. Now at this point, when we have fraction C, we have a very high concentration of ammonium sulfate. We have to remove this ammonium sulfate for the next step of protein purification. And what we are going to do for this is put this into essentially a beaker of water. But first, we are going to add the water here, and we are going to add a bag with clips on the end to seal the bag off. This is called dialysis tubing. There are very small holes, pores, in this bag. These pores are smaller than the protein, so the protein can't escape the bag, but they are large enough for the ammonium sulfate. So if we have a high concentration of protein and ammonium sulfate, by natural diffusion, the ammonium sulfate will move into the water. And over time, what we will have is called an equilibration of ammonium sulfate in the bag and in the outside when it reaches equilibrium. Then we can remove the bag with our protein in it and move to the next step of purification. Now the next step we're going to talk about is a little bit more specific, and it's called liquid chromatography. Liquid chromatography has several different chemical formats. The first one we're going to talk about is ion exchange chromatography. Chromatography is typically done in tubes, long glass tubes. You add the sample from the top. The sample percolates through the column and then elutes out the bottom, and you collect in a tube. And this is called the eluate. Now what makes liquid chromatography unique is the material inside the tube that the protein sample is percolating through. This material is called a matrix, and the matrix can differ for different types of liquid chromatography. So one type of matrix is ion exchange. In this case, for example, the matrix consists of beads. And these beads are packed inside this tube for fractionation. These beads, for example, are negatively charged. What will bind to this are positively charged proteins. Any proteins that are negatively charged will flow through unbound, and they will come through first, whereas the positive charged proteins will bind to this negatively charged matrix. So again, recall that these types of proteins have very high isoelectric points. They're positively charged. Once the negative charged proteins have eluted from the bottom, they're now separated from the positively charged proteins bound to the matrix. We can release the positive charged proteins by increasing salt, for example, sodium chloride. High concentrations of sodium chloride, sodium plus chloride minus, these sodium will bind and displace the protein from the negatively charged beads, and the negatively charged chloride ions will shield the positive charge of the protein from binding back to the beads. And then this will elute separately, and we've separated the positive from negative proteins. This here, because we're talking about positive proteins, this is called a cation matrix, or cation exchange, because positively charged molecules are binding to the matrix. The reverse principle is also possible using anion exchange. In this case, the matrix beads, which are these, are positively charged. So we have just the opposite case. Here, with anion exchange, the negatively charged proteins will bind to the matrix, and the positively charged proteins will flow through. The negatively charged proteins that are bound to this matrix can be eluted exactly the same way as the cation exchange with salt, displacing the charge interaction between the protein and the beads. This is called ion exchange, either cation exchange or anion exchange. The next type of chromatography is also called size exclusion chromatography. Once again, we have a long glass tube where we have sample entering the top, and we have eluate dripping into a tube to collect, and the sample will percolate again as before through the column. But size exclusion works simply on molecular mass rather than charge. Ion exchange works on charge. Molecular mass is going to explore the differences in size between proteins. So the way this works is we have beads that are once again part of the matrix that is within the column. But these particular beads do not have a specific positive or negative charge. What these beads have are holes. And these holes are of a very precise size. Small proteins will enter the bead because they fit through these small holes. Large proteins will not fit through the hole, and they will percolate between the beads because they do not fit in the hole. Now imagine a lot of these matrix molecules, these matrix beads, in this column, and as small proteins are percolating through the column, they are entering and exiting these holes. The large proteins are going around these matrix beads. So over time, when you have a mixture of large and small proteins entering the top of the column, the small proteins are going to take longer to go through the column because they're going in and out of these beads. Large proteins can travel rather rapidly because they're going in between the beads. So what this means is that the first eluate coming out of the column will be represented by large proteins. Later, the later eluates will be small and medium-sized proteins that have percolated through these beads. So this type of chromatography takes advantage of molecular mass. Different sizes we can separate. The last type of chromatography is called affinity chromatography. Once again, like the other types of chromatography, we are going to have a column with sample entering the top of the column and eluate being collected at the bottom. We are going to rely on beads once again. The beads will be the matrix. And these beads will be in the column, and our sample will percolate through them. But for affinity chromatography, this takes advantage of the enzyme substrate binding parameters. So if we know the substrate of our enzyme, we can take this substrate and covalently attach it to the beads. So with the substrate loaded on all of these beads, being the matrix, as the enzyme is passing through, it is going to bind specifically to the substrate because it has a high affinity. And this is where the name affinity chromatography comes from. So proteins that do not bind, do not recognize the substrate, will flow through. Enzyme that recognizes the substrate will bind. And simply to isolate the enzyme, all we have to do is displace or dissociate the enzyme from its substrate bound to the matrix. This, of course, can be done by disrupting the bonding energy between the enzyme and substrate. The typical way to dissociate enzyme and substrate in these is to increase salt to disrupt ionic interactions between the substrate and enzyme, or decrease the pH. Oftentimes decreasing the pH, making it more acidic, will protonate groups that are responsible for charge interactions between the enzyme and substrate. That will displace it. Also, mild pH changes, mild acid conditions, will just slightly denature the enzyme, unfold it just enough so it releases from the substrate, loses its affinity. And then once it comes down here into the eluate, we can return the pH to 7 neutrality to protect the protein from acid denaturation. So these are the major types of purification, enzyme and protein purification that we use. And now how do we quantify, how do we measure the amount of protein purification for each step that we've taken? So starting from ammonium sulfate and then progressing through our various types of chromatography, we need to measure how much enzyme, one is how much enzyme we are recovering from the specific purification step, and second, how much non-enzyme protein are we eliminating? So during protein purification, what we want is pure enzyme, single molecule that we're studying. We need to remove all other protein. So we need to measure the enzyme we're recovering and measure how much of the protein that we're not interested in we're getting rid of. So what we are going to do is apply what's called specific activity measurements. And this is done in all protein purification schemes when we're examining an enzyme. So let's start out and say that we have a crude cell or tissue extract. So this is our starting material that we've obtained from sonication or detergent or shear force. And let's say we measure how much total protein is in there. and we are going to find there's 10,000 milligrams of total protein. Now in this extract is our enzyme. So we can measure the enzyme by mixing the specific substrate with the enzyme and measure product formation. Let's say that we have, in this initial extract, 100,000 units of enzyme activity. Then our specific activity is going to be defined as units per milligram of protein. So we take our units, divide by our total protein amount, and we have 10 units of enzyme per milligram of total protein. Now let's say we do our precipitation with ammonium sulfate, and we isolate that specific fraction that precipitates at the precise concentration to eliminate non-specific, undesirable proteins and obtain ours. And we take that, we go through dialysis to remove the excess ammonium sulfate, and we measure again the total amount of protein in that dialysate. Let's say it's 3,000 milligrams total protein. So you can see that we have lost a lot of protein here by this one step. Now if we measure our enzyme, let's say we have 96,000 units of enzyme. Yes, we have lost enzyme in this step. Protein purification is not perfect. However, if we calculate our units per total milligram, we see now we have 32 units of enzyme per milligram of protein. This represents approximately threefold purification. So even though we have less enzyme, it is more pure because we've removed some of the contaminants. Now let's say of our next step, we do an ion exchange chromatography step. And the solution, the eluate that comes out after eluting the column, we have 400 milligrams of protein left that was bound to the column. And in here we measure 80,000 units of enzyme activity. If we calculate our specific activity, units per milligram, we see now that we have 200 units per milligram of protein. This represents greater than sixfold purification over the previous step. Now let's take our protein purification and go through one more step. Exclusion, size exclusion chromatography. We take the material coming out that has our enzyme activity, we measure the total protein, and now we see that we are down to 100 milligrams of total protein. Again, we've lost enzyme activity overall. However, our specific activity continues to climb. This indicates that our enzyme is getting purer and purer in the solution of protein. Now finally, let's assume that we understand the substrate for this enzyme, and we set up affinity chromatography so that the enzyme binds specifically to the substrate. After we elute the column, 3 milligrams of protein are left. Enzyme activity, we measure a total of 45,000 units. Calculation of specific activity gives us now 15,000 units per milligram. So we've seen from one step to the ammonium sulfate we have a threefold purification. The next step gives us an additional sixfold purification. The next step is a threefold purification. This step here is approximately 2.5. Overall, You take your final specific activity, divide it by the original, and what we have is a 1500-fold purification of our enzyme over the original material. And this is how you calculate specific activity and quantitatively measure the effectiveness of your purification scheme for that protein or enzyme.